Hello, everyone. I would like to welcome you all for another episode of FCPI Podcast series. So far, we have been covering various topics relating to clinical pharmacy and practice relating to clinical pharmacy. So today's episode is all about emerging antibiotics and their clinical application. Thank you for accepting our invite, Dr. Maneesh Paul, sir. From your busy schedule, giving your time for us for the podcast series is only the biggest thing. Thank you so much, sir. So, with no further delays, I would like to introduce our guest for today's episode, Dr. Maneesh Paul. Uh, Dr. Maneesh Paul is the co-inventor and program lead of the first invented in India anti-infective molecule enmetazobactam at Orchid Pharma, Chennai, India. That has been approved by US FDA and UK MHRA, European Medicines Agency, and Indian CDSCO for marketing authorization in 2024. He is a clinical microbiologist who pursued basic and applied research discovering novel anti-infectives and characterizing several microbial genes and proteins. He has translated scientific accomplishments and leadership through patents and publications. As a postdoctoral fellow, Sir is trained in infectious diseases, specifically in molecular mechanisms of the pathogenesis of neonatal microbial meningitis at Johns Hopkins School of Medicine, USA, as well as the role of actinomycetome commitants in the pathogenesis atherosclerosis at Umeå University, Sweden. He was the Orais Fellow at Center for Biologic Evaluations and Research, US FDA researching on reserve vaccinology of Neisseria meningitidis, and as an industrial fellow in anti-infectives, new drug discovery program at Dr. Reddy's Laboratories, India. Dr. Maneesh, in addition to leading the enmetazobactam program, also led anti-infective discovery research group in India, co-inventing several preclinical candidates in antibacterial and antifungal space. He led transdisciplinary research at Acharya Institutes, Bangalore, enabling it to become a research-based academic and research excellence campus. In 2022, Dr. Maneesh founded Microbioma India, a research-based organization founded on varied science to participate in addressing the challenges of AMR and WHO's One Health mission by providing microbiology-based solutions and stewardship initiatives. Dr. Maneesh is an AMR stewardship champion through AMR Action Collaborative Engagement. He served as a member of the AMR Committee of the Infectious Disease Society of America, USA, for 3 years and as a member of its research committee. He conferred the qualification of Fellow of IDSA in 2024. Dr. Maneesh received master's degree in medical microbiology from Kasturba Medical College and obtained a PhD from Gulbarga University, supported by Jean Claire Lee of Harvard Medical School, Boston, USA. Thank you, sir. That is the longest introduction.
I think we have taken most of our time in introduction. Thank you, Dr. Nischala, for this introduction. It's a pleasure to be here in this podcast. It's not that my busy schedule or this elaborate CV that you were introducing me to. It's more of I enjoy me enjoying the sharing of whatever little knowledge we all have acquired along our paths. And at the same time, I would encourage more people to speak up if there are audiences to let me know if there are any questions they would like to ask me. And it could be more interactive because learning happens mutually always.
Yes, sir. Definitely, definitely.
Sure.
So for it to be started, like once I was going through on your LinkedIn, once I read your intro, so I was very curious to understand how the journey has begun from being a graduate to being an inventor. So can you just give a brief about it, sir?
Well, that's a totally personal journey of mine because I started when I was in, it's almost like a cliche when I say this because like most of the scientists, my inspiration came by uh, the age when I was in high school. It was a chapter by Louis Pasteur, which taught us about this invisible world around us. And I was like fascinated. There is a world around me, a life floating around me, and I don't know about it. So that made me curious. And I talked to my science teacher, Miss Priscilla, and she was like, okay, what do you want me to do? So I said, let me have access to the laboratory where I could go and explore, this microbial world. And she did let me, and that's when I started using the microscope, the compound microscope, the light microscope, right? And then that's where my interest came in. And I thank my parents for not pushing me to become a clinician, which was this, it was like a stereotype. Everybody either engineer or a
A
clinician, only
doctor.
two options.
Yeah.
Yes, that's the two things. And I said, no, I don't want to be that. I want to become a scientist. And I put a plaque with all the scientists in that one, and I left one column open. I said, one day I want to be there. That was my
Oh,
vision. Yeah.
that's so great, sir.
So from then, of course, I migrated into clinical microbiology, was practicing, and then I realized that my niche was in more of inventions. So I went and trained myself as a postdoc at Hopkins and at the USFDA and UMI University. I came back to India and joined Orchid Pharma, where we invented this molecule. So it was pretty much a vision translation into action. And I really thank my God and my parents, everyone, for having this support and guiding me in this direction. And determination comes from within always, and some spirit within you has to drive you for that.
Yes, definitely, sir, definitely. Uh, it was like for everybody who are in the graduate programs, like everybody will dream to have that sort of career, and then they sort of like in the journey they then deviate into various paths, what they have decided and where they have landed. So that's how the stories will be there. But what you have decided to be, and then you being there at the final destination is the biggest thing, sir.
It's a blessing.
It's, yes,
Yeah,
it's totally a blessing, sir.
thank you.
So like you have been seeing so much, like when you have started your journey about the antibiotics, the usage, the resistance, which is now growing, and it had become like a global concern. Could you share your perspective on how these newly introduced antibiotics or the global resistance, how is it going to help?
There are several aspects to it. Resistance doesn't develop that easily because it is already there. We always tell the most wisest living organism on this earth, which we know of how, or have encountered, are the microbes. And they know everything. Every structure we want to talk about, they are pretty much aware of it. So there is a rising threat of untreatable infections due to multiple drug resistance or extremely resistant, organisms with pan-resistant pathogens coming along the way. Ones which were curable infections have become quite risky, including pneumonia or sepsis or urinary tract infections. All are becoming life-threatening. You must be experiencing this in the clinics as well, especially in the
Yes.
ICUs.
Yes.
There is higher morbidity and mortality with these patients.
Yes.
So WHO then warned us that AMR could push us back to pre-antibiotic era. The reason being, uh, if not for that, they also coined the word the silent pandemic to this, right? So it's a pandemic existing there, and we are ignoring it, and it requires a lot of mobilization. That's the reason probably what you see on my LinkedIn profile and everything. What I write is to sensitize people that this is something you cannot ignore. And the second reason is exhaustion of the existing class of antibiotics. See, most of the antibiotics that we are using today, they are like invented somewhere between 1940s to 80s, right? After that, we have been only improvising the molecules. But the gram negatives are the ones which pose the greatest risk for us because they have multiple mechanisms they can adopt at the same time. They don't want to use one mechanism, they want to use dual, triple mechanisms.
Yes.
And that is leading us to use of carbapenems. And when we use carbapenems, obviously carbapenomases will come up like NDM and OXA48, KPCs. KPCs were not known in India, earlier. Now I think that is not too far when we start seeing more of KPC,
Yes. Yes.
organisms. The other thing is, of course, the innovation gap, because in innovation, not much money is being put.
Yes.
Most of the companies have withdrawn because of the economics. I say economics because the ROI is very low. If you, as a clinician, you would prescribe when a patient comes with metabolic disorder or cardiovascular conditions, it's a lifetime medication. So the pharma company continuously reaps the benefits of sales of these molecules. For anti-infectives, 3 days with azithromycin, two you've got maybe 14 days of intravenous drug. So where is the ROI on it? So economics is very, very, very bad for this. And in top it to that, we have now with AMRs towards like me and you and many, we are trying to say that restrict the use of antibiotics. So
Yeah.
where is the money to
Like
invest into R&D?
definitely.
So these are some of the reasons. And of course,
Mm,
there is a lot of impact on the global health and economics. If the
mm,
death rates increase, what would happen,
mm.
right?
Yes.
In the marginalized countries, third world
Mm,
countries, it's going to take a toll on everybody. LMICs especially is a very, very tricky thing. So I would look at critical role of modern medicine in this. So we when you have AMR, the resistances, when we talk about,
mm.
look at patients with organ transplants or chemotherapy or dialysis. How do they go about if you do not have good antibiotics to save the patients? Because
Yeah.
all of them rely on antibiotics as a prophylactic now. So you
Yes.
dose a linezolid, an oxymolic,
Mm,
oxazolidine molecule, or you dose any of the higher class of molecules in the beginning of the surgery to prevent. So
mm.
instead, if you have good hospital infection control practices already in place,
Yes.
we would prevent infections from coming in. Because yeah, our body is very, very, very robust. And we
Mm.
have to let it fight it as well. But not, of course, in
Yes.
surgery or immunocompromised patients, but otherwise, normal patients, we should encourage that. And then the last one I
Mm,
would like to touch upon would be uh, time sensitive opportunity.
mm.
See, new antibiotics are not just replacement drugs,
Yes.
right? So we have to have proper diagnostics,
Mm,
proper stewardship initiatives, proper infection control prevention activities. So now, without these things, innovations happening now, future generations may inherit a world where minor injuries also can cause
yes.
death.
Resistant organisms.
So it's very important to understand this AMR in this context, that it's not a joke. It's not something
Mm,
we can turn a blind eye and say, okay, we don't have it, so we don't experience it, so let's be cool about it. No, it's not cool about it.
mm. So it is not just about one person being concerned about the AMR or one specialty being concerned. As a whole, it is like the combination of everybody, the general public, be it the medical fraternity, everybody have to be concerned about AMR. That's what
Absolutely.
I feel. Yeah.
And also, people have this wrong concept to say that, oh, pharma industry, they keep making money, so they just want to sell
Mm,
antibiotics. No, it is not
yes.
that they want to sell antibiotics. When it has so
Mm,
such a low revenues, why would they put so much effort into selling that rather than putting their efforts into some other lifestyle disorder,
mm.
antibiotic, I mean, drugs.
Like if we see any other industry developing a new molecule, like when we consider antibiotics or antimicrobials, we see very few drugs which comes into the market in whatever speed that we see. Like very few come into the market, and it takes longer time too. So what do you think?
See,
So,
microbes are smarter, as I told you, right? So
mm,
if you bring up one antibiotic, it is just the value of t, which is time,
mm,
how big or short they will decide. Microbes are highly, highly collaborative. This particular forum you have set up now,
mm,
what is the reason you're putting this forum? You want your audience to get access to whatever information that is stored within me, which can be revealed to everyone in a larger platform like this. Otherwise, only a secular, maybe 50 or 10, 20 people are going to sitting and listening to that knowledge. So like this, there are so many people who are sharing knowledge with your community of people. This is what you're doing, what the microbes do, sharing the information. You're like a microbe
yeah,
now, right?
yeah,
You're doing that job of a microbe of,
yes.
spreading the word of knowledge of
Mm,
antimicrobial resistance or whatever multiple areas of your domain, what you talk about in your forum. So microbes, when they realize that there's one new drug has come in, they have an R&D center opened up instantly. It all goes into the R&D center. They analyze and say, okay, humans have come up with a new molecule. It is called gepotidacin. Okay. Okay. This is it is used for complicated urinary tract infections. So now, or uncomplicated urinary tract infections. So I'm going to beat it now. So what do we do? So they all come and have a conversation, quote unquote, and then they come up with a mechanism to overcome it, because this molecule was predominantly made because it inhibits bacterial DNA gyrase and topo4,
mm,
okay,
yes.
through novel mechanism. So we have humans have thought through so much and tried to see that, okay, we can bypass it. We can
Mm,
prolong the time before resistance develops. So
Mm.
such new antibiotics are there, but the first-in-class antibiotics reaching the clinics are very, very difficult because two reasons. One is the WHO's aware classification will make sure that the drug is kept for reserve category so that it could be used as a last resort. So that is one, to prevent AMR from coming into play. And the second is the cost. When you keep it that way, who would pay for my R&D and for my manufacturing, quality and everything? Somebody has to pay for it. So the cost will go high. So the access becomes another challenge. One is the restriction of the reserve category, and then no access to antibiotic. Access means not that it is, though financially somebody wants to fund it, it is not reaching to the right patients, right drug has to go. That is called access I'm talking about. Right? In India, access to antibiotics is very much there. He's just a pharmacist.
Yes.
He or she would just dispense whichever you give it, they will dispense. So I'm talking of that access, the real access of antibiotics to those patients or the deserving patients. That is the access. And there are some antibiotics coming up, quite a few. One of them, you're talking to the guy here, the N-metazobactam. It's an
Yes.
antibacterial drug, right? But of course, this came last year. We launched it and approved rather. But the invention happened some time ago, which I'll talk to you a little later about how it went about it. But this is a class wherein the beauty of this molecule is it is like tazobactam. It's a beta-lactamase inhibitor. So our approach has been that, without triggering a newer mechanism, existing mechanism, how can we become more potent? So the IC50, what you call as, the number of molecules required to bind to the site, we reduce. So what happens? That means when the enzyme is there, the number of drug molecules which go and bind are very few. That means it's highly potent. When it's highly potent, what happens? The turnover number, what we call the number of molecules which go and come inside, are going to reduce. And the binding is tighter. So it is much more potent. So that way, what happens? We are extending the life of the antibiotic, which is given along with the FCPI, or it could be piperacillin, whichever drug you want, beta-lactam. So this is another method how we are trying to fight. One is a novel method, which I mentioned a little ago. Now, Roche has come up with another molecule, right? So cerebalpin, you must have read about it quite a bit. So there's quite a few molecules are coming, and people are also coming up with newer combinations with astronam and
Yes.
yeah, all this,
Avibactam.
because astronam it's a monobactam and it's got its own stability. And when you combine it with the Novoxel molecule NXL104, the new name is I don't get it right now. But anyway, so it is a combination drug, right? With NXL104 was its original name. Now I'm not getting its name itself. That's short term. But anyway, NXL104 was its original name, which is a very popular Avibactam.
Avibactam, yes, sir.
Avibactam's original number. As a scientist, we remember the molecule numbers, not the commercial name so much. I know the NXL104 was its now, the ID. Yeah, so we have quite a few of these kind of molecules which are coming. So instead of attacking the molecules directly through novel methods, we are also trying to restore the earlier antibiotics. There's a beautiful paper which was written by a couple of senior authors such as like Karen Bush or Matthias Barker. They all written on how to bring back the older molecules. You must have heard of molecules such as, I don't think you ever heard of apalcillin.
No, sir.
No, right? That's an amazing molecule, but never reached the market in globally. When I look at that molecule structure and its spectrum, it is amazing molecule. For whatever commercial reasons, they did not make it to where it had to come. So this way, we have quite a few of older antibodies they have captured in the review. So if we pick from there and bring it back to life, that's also a good thing. But who's going to fund it?
Yeah.
Funding.
Yes, sir.
So yeah, new classes and mechanisms are very important, and because they provide options for us where resistance
can be
is compromised.
targeted. Yeah. Yes, sir. So as you were telling about the general public, how we have access to all the antibiotics in the pharmacies. So what do you think, sir? How can we restrict these antibiotics to the general public? Or do we have to bring in awareness or do something about it?
It's a very loaded question for a forum such as this. So I'm going to be very balanced in my emotions. It really ticks me off at the way we are dispensing antibiotics.
Yes.
Starting from the drug regulators, they are trying their best.
Mm.
Government is, announcing initiatives to control that. But it's a very tricky situation because
Mm.
now see, when a patient comes with an infection, the right method to treat the patient is the patient has to be evaluated by a clinician, and then you have to diagnose. How do you diagnose? You do a laboratory test investigation, and then you say, okay, this is the antibiotic you have to give. Now, what is the cost, both in economically and also time?
Yes.
So at least 3 days has gone by. And
Mm.
by the time, most likely, if it's a milder infection, patient would recover on their own. If it's tougher infection, then of course it will need further intervention with the clinician. But the cost, financial cost, is so high.
Yes.
So what would a common man do? And at the same time, the one more aspect to it is the ratio of clinicians versus patients is highly
Yes.
skewed in India.
Highly burdened.
Because we are a highly populated country. So
Yes.
that's the way it is. Nothing wrong with that one, right? So now what the patient does is, instead of doing this, first time he or she would have done this whole process and spent more money. So they would straight away walk up to this guy and he will say, I went and asked this question myself. Hey, I'm having some throat issue. Can you give me something for me? He pulled out Augmentin. Okay. And he gave it to me. So I turned it around and said, this will work. Yes, sir. You take for 3 days, you'll be fine. There is something called red line. No, they say red line means you should not take it. He said, sir, some antibiotics we can give, sir. Augmentin, azithromycin. He's telling me directly, and I'm like, okay. And I just walked away because I didn't want him to get scared now. So the point is, they think they're authorized to dispense antibiotics. So education of pharmacists that they should not dispense antibiotics, and they should not play the role of clinicians there, but rather counsel the patients that wait for 3 days with your with your fever, you will be fine. Use our Indian methods of jadi booties. There are so many kashayam, gishayam. All those things boost up your immune system to fight because we have got inherent ability to fight infections.
Yes.
But in this fast-paced 20-second reel watching, time span, we don't want to wait for 3 days to get cured. So at one of the talks I was giving on WHO's annual day, I was mentioning, when a child becomes sick, you have to first get an anti-anxiety medication. You give it to the parent.
To the parent, definitely.
Because parent is so anxious. What happened to my baby? And they rush. They force the clinicians to prescribe antibiotics also.
Yes.
But today's world is changing. I must say, a lot of parents have become aware. They are telling, no, we will wait.
Yes.
So it's all about education. So to address or an answer to your question, to address the challenge you posed, is it's not only to blame any regulators or pharmacists or the government systems, but education of the common man. So I wear a badge, either it is a Sustainable Developmental Goals or an AMR badge. So this is a conversation starter. People will look at it and say, okay, what is this about? Then I'll, preach.
Start talking about it.
Definitely. So this is the way we must, everyone who's aware, if you start any social gathering in a metro or bus on the way, in a social gathering, keep educating more and more people. And they should become ambassadors. This is what I do in AM race, antimicrobial resistance, action, collaborative engagement. One person cannot do this. It's a collective. All of us put together can do this.
Definitely, sir. Definitely true. That is how the start of everything will happen. Otherwise, if everybody switches the responsibility, it just goes off in air.
Yeah, and also, when you use broad spectrum antibiotics, I am very skeptical when it comes to broad spectrum because they do affect our gut microbiota, which are playing a very important role in our immune system. So I'm a fan of narrow spectrum, pathogen specific, pathogen focused, right? But commercially, that is a very tricky thing, right? So for a clinician to prescribe, clinician would say in India, if you go, I prefer a broad spectrum. I do not know exactly. I don't believe in the microbiology report.
Yes.
Because it is not correlating with my clinical picture.
Yes.
Right? Because microbiologists, I won't blame them also because the whole exercise starts from sample collection. If the right sample was not collected the right way, the whole exercise following that is no use.
No
So
use.
it starts from there. So these
Mm,
nurses or the laboratory technicians who are going to collect at the
mm,
from the patient, they are overburdened. So they'll just
mm.
wound swab. It's just usually skin swab. So
Yeah.
you isolate.
This, yeah.
How many of
Deep
them collect?
seated, they don't take it. Yeah.
So this is where I think education of the our medical
Yes.
fraternity
Mm,
and the pharmacists would definitely contribute in controlling antimicrobial
yes.
resistance from becoming more stronger than what we are
Yes.
anticipating.
Yes. Definitely. True.
Yeah.
So, with these emerging antibiotics, and like you've mentioned, there are many molecules which do not come into the market. They end up in the clinical trials, or for whatever reasons, they don't come into the market. So, what would you say about it? So, because you had been in that journey, so if somebody like a person who is starting the journey with a new molecule, and what could be the reasons that they couldn't get into the market?
Well, there are several reasons. I think I did allude to these questions in a way already because of this aware classification, the stewardship,
Mm, mm,
right? So many go into the reserve category. If a
yes,
broad
yes,
spectrum antibiotic comes in, the likelihood of it becoming reserved is very high.
mm, yes,
Uh, on the other hand, a molecule like N-metazobactam, which I spoke about, which is our invention from orchid, it can become an access molecule because it is not directly killing the bacteria.
yes,
It is attacking the bacteria's resistance mechanism,
mm,
which is the beta
mm,
lactamases.
yes,
So, and there's one more zidibactam from
mm,
Wockhardt, right? It has
mm,
got dual
yes,
action. It is not a BLI. It has
mm,
got a beta lactamase inhibitor activity, and also it has got inherent antimicrobial activity. So
mm,
in combination, cefepime, it's a very potent one, but likelihood
yes,
of it triggering a newer mechanism is, I feel, is low. So
mm,
that's another good one. So
yes,
it is. I mean, these are some of the reasons why you know uh, mm, access is becomes a challenge. How do you decide?
yes,
So you need to decide on access. For each country, you need the right kind of data of surveillance. India has
mm,
started now. Earlier, we had only 11 or 12 centers. Now we have gone to
mm,
double digit, triple digit of surveillance centers in the country, which is going on.
mm,
But the diversity of population we have, the diversity of food habits we have,
yes,
the diversity of religious practices we do based on which we
mm,
eat certain foods,
yes,
all these things modify our microbiota. So
mm,
the way we respond to infection, each of us differ
yes,
quite a
yes,
lot. We are not like
mm,
one brand, like, okay, we're all, of one descent, one kind of population. We cannot classify. India is
yes,
highly diverse. So in
um,
a country such as India, the local clinician in that area knows, okay, now is the season for this. This is what
mm,
I'm going to do because they have this on the go acquired knowledge, what they use
yeah,
and make it happen. So I think
yes,
in India, the way we have to address stewardship is it, it's very dynamic.
mm, yes,
But
yes,
newer molecules are becoming difficult because most of the resistance arise because pathogens have already, quote unquote, seen decades of exposure to existing targets,
yes,
like be it penicillin binding proteins,
mm,
ribosomes. So
yes,
we have to use newer binding site mechanisms.
mm,
That,
yes,
because then that's why what happens, the pre-evolved defenses are not there in bacteria yet. So they have to
mm,
learn, they must go back to the bench. So
yes,
that is the time we get to take care of this infection. And other thing is I would recommend is antibiotic cycling also. Right?
mm,
Antibiotic cycling is a good method because what microbes are single cell and they don't want to
mm,
waste their resources. They're
mm,
highly conservative. So what they
yes,
would do is when if penicillin binding protein targeting antibiotics are removed from the system for a while, they would revert back to their normal self because there's a lot of stress to the microbe to
yes,
survive in
mm,
an artificial structure which they're holding onto themselves. They want to
mm,
unwind, right?
mm,
So
yes,
that time we target the other mechanism. So their focus
mm,
goes towards that. Then again bring this back. So that way we can
yes,
gain time between treatments. And that's one
yes,
of the reasons I think we have this multi-drug treatments, right? We use
yes,
with the BLI, we use as a aminoglycoside.
yes,
That's the reason we
mm,
do those things.
mm,
Yeah.
yes. So, bringing back to the molecule that you have invented, sir.
Oh,
So
yeah.
how that can you take us back to the moment or when you had that idea of for N-metazobactam and how the journey had begun?
Okay, it was actually a strategic decision by the organization. Okay,
Mm-hmm,
Orchid Pharma has been in drug discovery for a long time. I mean, in pharma industry since 1992, an
mm-hmm,
amazing R&D center. So we had the new drug discovery program where we are working on new oxazolidinones, new cephalosporins. We had HDAC inhibitors. We had like multiple targets we were working on, right? For MRSA, candida infections. We are working on multiple targets we are working on. And we know that the journey is quite long for novel molecules, and the likelihood of it becoming success is lower due to the resistance development. So that's the time we had internally already being a in a manufacturing company, we have process R&D, what we call it as, right? Where we improvise the processes and keep making newer process. That's how we generate revenues in the generic market. So now what the decision was made at that time by the then scientists was, why don't we start a low hanging fruit? So we'll get into beta lactamase inhibitors. Why don't we improvise tazobactam? So that is when the R&D started with. So what the chemist did was Dr. Senthil Kumar led the team there, and he's a brilliant process scientist. So he started making with his team the one side chain shifts because he's a patent head as well. So he could find, okay, this is one space which is not covered in the patent. Let's explore. They started making molecules. So once the molecules come into play, the molecule is just as a molecule unless life is given to it. And it's a biologist whom all the chemists hate. The reason is every molecule they come up with will say it is inactive. They would have taken 6 months to 1 year to make one molecule, and in 24 hours will say it is inactive. Look at that. Tragic, quite tragic for them. So they hate, quote unquote, hate biologists, but we all love each other. It's like a family. So when this molecule was known as OCID 5090 and metazobactam earlier. So this molecule did show in vitro activity, but there is in vivo. There is no in vivo activity. When there is no in vivo, this molecule is just goes to the shelf. Just like this program was not going anywhere, essentially. That is the time I came into the picture. So I was like, okay, if the standard method doesn't show, we are discovery, we are scientists, we need to create a newer models. Why don't we create a newer model? So I came up with a very different approach. So how do you do it? You have a known molecule, right? You take the known molecule and show the efficacy of that known molecule in the new model. If it is as expected, that means this new model is validated. So I developed a newer model. And then when we compared that along with my molecule, with the N-metazobactam now, the efficacy was like boom. So that is when this molecule got life. That is the birth of the molecule. Until then, the molecule was there. It's not developing. It's maybe in the morula stage. Just there, fertilized, but sitting there, nothing happening. It can implant or it may not implant. When life comes in, implants. So I call myself the mother of the molecule because it was 18 years, means gestational periods in years to carry the
Yes.
molecule, to bring it to the molecule, to the drug, right? To bring it to the
Yes.
drug. So that was the turning point for this molecule. Without that's what I published in the paper as well. The invention of N-metazobactam in American Chemical Society. I wrote this how this turned the path. This is when the actual invention happened. After that, it's developmental. We have a lot of scientists who came who tried to improvise the process for this, and you can see that for development we can do in-house or we can outsource it. But our other biology programs were going on. We have oncology, CNS, metabolic disorders. We had all these programs. So we had an amazing pharmacology department. So they developed, there was one gentleman called Baswaraj. He was very much involved in developing a formulation because bioavailability is important. So that's when he became. So it is not like, that's the reason I call myself the lead co-inventor. Lead co-inventor because I carried the molecule from the time it got the birth till until now. But others all have contributed significantly. Without a good formulation scientist, my molecule would not get bioavailability. right? If
Yes,
somebody
yes.
doesn't develop a good PK model for me to show how its distribution
Mm,
is happening. But one thing I'll tell you,
mm,
there are three aspects of it: a good synthesis,
mm,
a good translation into in vivo. Now, for
mm.
these two things, you need chemists, you need pharmacologists, you need
Yes.
pharmacists, all those people work together. If the
Mm,
formulation is good,
mm,
and moment it translates to in vivo, rest all is only
mm,
decoration. It's
mm.
you're doing everything to explain now.
Yes, yes.
Invention is the moment it got the life, the drug is
Mm,
done. It's that we have
yes.
invented.
Mm,
Eureka, we have done it.
yes.
Now, then we have characterizing is what we do. And then we
Mm,
optimize it for human dose. So
mm,
that is all next stage of it, developmental
mm,
activities. So that's why we call it as R&D is discovery and development.
mm,
Invention happened during discovery and then the developmental phase starts.
mm.
That's how the whole process. Yeah, that's the moment when in vivo translation happened. And I had an amazing team, Dr.
Yes.
Hariharan Periaswamy, who was with me that time. Now he is part of
Mm.
this IDVACTM program at Wokhart.
Oh,
So he's the same guy
great,
there
great.
as well. So he
Uh,
did an amazing work in he was an amazing, he's a veterinarian who
mm.
thinks very well, and he would do amazing work with me.
Uh,
So
it all
this,
happens with a great team, sir.
yeah,
Definitely.
absolutely. All these people have worked so well,
Yes.
pharmacologists and toxicologists. See,
Uh,
you need to see toxic, what is the level it can tolerate. See, once
yes.
in vivo has translated, no, all those things
Mm.
are to characterize the molecule and to see whether
Yes.
compliance has given certain standards. You cannot have more than this, all those.
Mm.
Does it comply within those limits? All this is compliance activities after that.
Mm.
So that's how discovery happens. It's a beautiful, wonderful journey.
Yes,
And
sir.
my whole team, I really, I'm
Yeah.
really fond of them because they are all work. Because once they saw this going forward, everyone
Mm.
put their heart and soul into this to make sure
Yeah.
this reaches the market. Yeah.
It's such an amazing journey, sir, to hear. Yeah. So just can you let us know from since the start of the clinical trials to getting into the market, what are the challenges that you had faced?
The first is, of course, once we have invented the molecule, it's pretty much done, right? We have done the job. And then
Mm,
what happens is commercialization.
mm,
After this was done, I wore a hat of a salesman. I used to carry my bag and go to all the conferences because the managing director,
mm,
Dr. K. Raghvendra Rao, he called me and he told me, Manish, if you think this can go forward, let's push it. Because as a
mm,
company, we won't have the financial ability to take it till the market. It's very expensive
mm,
journey. So we wanted to partner with somebody to take it forward. So the
mm,
one Prasad Patala, he was the business development guy. So he
mm,
used to go and find the leads and present to them already what the program is about and get them interested. And then I would go there and start pitching for it. So it's not
mm,
one company. I pitched to many big pharma, small pharma, startups, and everything. And one
mm,
gentleman by the name Nicholas Benedict is the first guy. He said, "You know what? I like this molecule. Let's look at
mm,
it." Many of the big
mm,
pharma were like, it's a useless molecule, oh, no potential and all. The reason is
mm,
probably it could be, interfering with their own internal programs as well. That's
mm,
understandable, right? For
mm,
any
mm,
company, you want to commercialize because already the revenues are so low. And
mm,
when
yes.
something is there already, why you need one more? So that
Mm,
would be anybody would do that, of course.
yeah.
A couple of them did that, but that this gentleman came up and he said, "Let's form." Then he came in picture. Then we had,
Mm,
for developmental stage and for licensing and everything. Dr. Sridhar
mm,
Narayanan was very part of it, who is the
mm,
founder of FNDR.
mm.
So that's why the journey is not only science; it's business as well now.
Yes,
So
yes.
business. So I became a salesman, and then we
Mm,
concluded Then Nicholas Benedict and his team, Stuart Shapiro, Stefano Biondi, Omar Lalo. These are the four gentlemen who came about. They evaluated,
mm,
and
mm,
then they in-licensed it. And Orchid owned 20% of the company at that time, and with revenues. But then when Orchid underwent the corporate debt restructuring, our shares in the company was diluted, and we only held the uh, royalties. So that's
mm,
for the agreement. So now we were working in that, with the royalty mode now because Indian market was excluded. So we are conducting right now, I'm leading the Indian clinical trials for this
mhm.
molecule. that is phase four. Of course,
Mm.
it is launched in the market, but yet we need to do the,
Mm.
how does it behave in the Indian population?
Mm.
As of now, looking at the data, what since October it is launched and it is
Mm.
doing amazing. So the data
Yes,
also has
sir.
to come clean for us. I won't expect any red flags here.
Mm.
So that's good. But very interesting thing happened a couple of months ago.
Mm.
The company Allegra Therapeutics, who had in licensed our molecule, they declared bankruptcy.
Mm.
And Orchid was one of the bidder there, and we got
Mm.
to own Allegra Therapeutics now. That means the molecule became back home. So that was a
Ah.
day of joyous for me. I was like, wow, this
Yeah.
molecule has had such a turbulent journey from day one. It
Mm.
was rejected, but given
Yeah.
life.
Yes.
And then after given life, sold. After
Mm.
sold, lost the ownership connect. The owner
Mm.
is separated from the mother in a way.
Mm.
And
Yes.
then reunited back to the company, the mother company. And now,
Yeah.
so that's an amazing
That's
journey for Orchid Pharma.
yes.
From the,
Yes.
the promoters changed, okay, from Dr. Raghavendra Rao. Now it is Dhanukas who are leading it. And Dhanukas have not rejected this from day one. Day one. They always paid attention to this. Let us take this forward, Dr. Paul. And he would, and I like that spirit. That means this molecule has life and
Yeah.
it will save millions of lives, and it is doing already. In
Yes.
the market, it
Yes.
is there.
It
Yeah.
is. Yeah, even we had started using N-metazobacter molecule at our organization as well. So we
Amazing.
had been
So
seeing.
that see again when you come to the market side, right? Orchid did not have our domestic market, but
Mm.
for this molecule we have Indian rights. This is the only market we
Yeah.
had. This is where we
Yeah.
can recover some revenues. So we
Yes.
tied up with Cipla. So
Yeah.
Cipla became our partner there. So Cipla is already
Mm.
existing market. So
Yes.
they have
Yes.
two brands called as ESBLO SIP and SIP
Mm.
and MET. SIP and
Mm.
MET, Cipla and Metazobactam.
Mm. Mm.
I think. And then Orchid named it as Orblicef because
Mm.
they labeled it as XBLFEP.
Mm.
XBLFEP means extended spectrum beta lactamase inhibitor with cefepime, FEP.
Oh.
XBLFEP. So Orchid,
Uh,
we call it as Orblicef because
uh,
Orchid's beta lactamase inhibitor with cefepime.
that's how the name had come to the
Yeah,
brand.
that's our name, brand name.
Uh, so how did you name this N-metazobactam to the molecules?
Okay, that is because we have one methyl group right there.
Mm.
That is why that Me has come.
Oh, oh,
Right?
oh,
It is
oh.
it's a tazobactam. Essentially, if you look at the
Yes,
molecule, they
yes,
both are like except one
yes,
side chain is methyl. It's
yes,
methylated.
yes.
It's a methylated
Uh,
tazobactam essentially.
yeah. Oh,
Yeah. So
that's
we
nice.
had actually chosen cefepime before we out licensed. We had a
Mm,
choice of either combining it with cefepime or with apalsalin because I like
mm,
apalsalin because apalsalin also has inherent anti-pseudomonas activity.
mm,
So cefepime also has got that. So I was like, that
mm,
these are two good combinations. But Allegra
mm,
chose cefepime as the drug of combination because it was their decision. Because
mm,
and they did all the three phases of clinical trials, and now
mm,
we own that company. So now Allegra is part of Orchid Pharma. So that I can proudly say we did the clinical trials in a way
yeah,
because it
yeah.
becomes part of our asset now.
Yes, yes.
We did not do it was Allegra which did the clinical
Mm,
trials. We are only
mm.
looking at Indian market.
Um,
Hmm.
nice. So, what in the medical fraternity or the science field, what are the innovations or the technologies that you are expecting in near future that could shape the next decade for the antibiotic development?
That's the plenty there. See, one of the thing is we must look at as I'm more tech savvy guy. The reason
Mm,
is I would like to explore new things which come in.
mm,
So now the new thing is AI/ML.
yes, yes.
The same Sridhar Narayanan has another company called as Peptris,
Mm.
which is AI-driven drug discovery company.
Oh, that's great.
Yeah, so there is from MIT there is AI designed molecule, novel anti-MRSA molecule,
Mm,
and also
mm.
it acts on gonorrhea scaffolds.
Oh,
So
mm,
people have started using generative AI models, which can design
mm,
resistance evading molecules by
mm,
learning bacterial evolutionary constraints. Because as
mm,
human beings, we can think to a certain level only, and we also
mm,
think based on our access to the amount of information we have,
mm.
right?
Okay.
So now when we use these technologies, right, they can crawl the web and get the right information, not right information, at least they'll bring together all the majority of the information which I might miss out and put it together. From there, we can start the journey to start
Yes.
brainstorming and looking forward. So it's got amazing. It compresses the timelines as well from
Yes.
years to months
Yes.
and opens, I think AI/ML is opening the second golden age for antibiotics.
Oh,
And second
right.
thing we need to look at is the precision antibiotics.
Mm-hmm.
Uh, because the way, as I told you already, like, even in India, the way each of us are so different.
Mm-hmm.
People say, in a family, they say, I am resistant to azithromycin. I said, you are never
Mm-hmm.
resistant. It's the
Mm-hmm.
monks are resistant. But your body, the way it metabolizes, it
Yes.
is not
It's very much
giving enough
different.
bioavailability in your circulation for it to kill
Yeah.
the bacteria, right? So that's what it
Mm-hmm.
means when it's not working on one person of the family, but not the rest of them. Maybe his liver is not metabolizing properly. There must be some other issue,
Yeah.
right? Or it's gut microbiota, or it's his immune system is not contributing to the treatment. So each of us have a different thing. So precision medicine is, when you say 500 milligram, I do keep thinking about this question, which nowadays it is coming in many of the forum is, why only 500 milligram? So that is
Mm.
a safe therapeutic dose we have fixed within the therapeutic
Yes.
window.
Mm.
But how safe it is to use less than that? Does it trigger
Mm.
antimicrobial resistance? So now
Yes.
what happens is, every drug that comes to the market, we have to
Mm.
ensure what is its resistance development threshold level, concentration, minimum concentration. It has to be there in the blood so that it doesn't develop resistance. So
Mm.
over and above that, you put a safety margin, and that's where you fix the drug concentration,
Mm.
change the drug in
Mm.
patients with those
Mm.
diseases. That is one way. Then we can think of multiple alternative methods.
Mm.
Phage therapy, that is also
Mm.
precision, because phage therapy cannot be given like in mass. You have to make
Mm.
it for each person. Then antimicrobial peptides, it's got its
Mm.
downside as well, but still, you know,
Mm.
immunotherapeutics, because these are all, what happens is it diversifies the arsenals beyond
Mm.
traditional class of antibiotics,
Mm. Yes.
right?
Yes.
Hmm. Of course, we are using many beta-lactamase inhibitors, metallo-beta-lactamase inhibitors, all those things. But it's very important to have a diagnostic, rapid diagnostic on
Mm.
the clinician's bench
Mm.
from the sample. I would
Yes.
love it, because
Mm.
many of when the clinicians come under the pressure of the patient to prescribe an antibiotic, a clinician can just take the sample in
Mm.
front of them, show that it's a viral
Yeah.
infection, not a
Mm.
bacterial infection, so you don't
Yes.
require an antibiotic. So the patient
Antibiotic.
also is convinced,
Yes.
right? So if somebody
Yes.
has a chest pain, which could be due
Mm.
to anxiety,
Mm.
is it heart?
Mm.
Chest pain could be anything. You
Yes.
start manifesting in this hand pain, all this. So
Mm.
psychologically, you start playing around. So when you
Yes.
have an infection, you say, "Oh, what would happen to me with this bacteria? That bacteria, a lot of AM." Then they Google, and Google will tell
Mm.
you
Yes.
could die in an hour.
Yes.
So they get petrified, and they're like,
Yes.
"Doctor, please give me an antibiotic." So
Mm.
in such situations, if you have a diagnostic kit there on his bench or her bench, they would just say, "Okay, you have this infection. Don't worry about it. You are going to
Yeah.
be fine." That itself acts like a placebo, because it's coming from an authority who is
Mm.
well versed in that domain, and they would feel peaceful at home, and then they
Yeah.
will recover very well. So
Mm.
precision therapies are very, very important for us. And I would also like to touch upon one particular thing is, which is very dear to me, is we have to focus now when it comes to stewardship angle, right? One health and planetary health.
Yes.
People have been talking about planet, one health. We need to look at
Mm.
planet as a thing. Because
Mm.
it matters, because antibiotic use in agriculture, wastewater discharge, environmental
Yes.
reserve, these are all the reservoirs of AMR.
Yes.
So biotech based, wastewater monitoring is
Mm.
important. Surveillance is important,
Yes.
right? So
Mm.
when, see, all this makes antibiotic development part of a broader resistance ecosystem strategy,
Yes.
not just
Yes.
drug drug
Doing.
fighting. We don't have to do
Yes.
that. We can do this, as I mentioned, no, collaborative effort. All this put together, we
Mm.
can do plenty more.
Yes. Yes.
Mm.
There's not one area that the antibiotics is being used, not just the human population. It is like the various places that has to be targeted in order to reduce the AMR resistance. Definitely, sir.
And
Definitely.
and also, when it is, I don't say it's only antimicrobials. the
Yes.
junk what we eat. Has anybody
Yes.
done
Mm.
of a burger, the
Mm.
constituents, what kind of impact it can have on your microbiota?
Yes.
Does it trigger some element of your host microbiota that
Mm.
could get sensitized with a new molecule which is not used as an
Yeah.
antimicrobial, but it has
Yes.
got a property of it because nobody evaluated it for antimicrobial
Yes.
activity. It is a
Yes.
it is an ingredient for food, but it
Yes.
might have antimicrobial activity. So if it is
Yes.
given in subclinical doses, does it trigger AMR there? So it's a very
Mm.
complex thing,
Yes.
right? So does it then eat right, exercise right,
Mm.
sleep right? So these are some of the things we have to do along with taking any other things like chocolates or your
Mm.
ice creams or junks, whatever you eat, soft drinks, all those things could be. Because I have seen a paper in which your acetaminophen or beta blocker, all these have got impact on your host microbiota,
Yeah.
which means we are altering our whole self
Yes.
on the microbiota level.
Yes.
But
Even the regular usage of pantoprazole,
yep,
even that is causing the disruption of the gut microbiota.
yeah. So we have to. So I always say, uh, protect the invisible and preserve the indispensable. And
Yes.
protect the means your microbiota, you protect them and
Yes.
preserve the antimicrobials as well. That's what is my caption on my website, manishpaul.com. I
Yes.
put that for
Yes.
that reason. That when they open that, that's what they see. Protect the
Yes.
invisible and preserve the
Same.
indispensable.
Yes.
Do you think your audience will be sitting for so long? It's almost like
Oh,
50 minutes.
yes, yes. Talking to you, I don't even like. I want to like continue the conversation. Talking to you, it is like so interesting that you've made the conversation, the journey of it, and the way you started explaining about it. It is like uh, like people will be bound to listening to the conversations, definitely. So, but definitely we will have to end this evening the podcast session. So before ending, I would want you to like give some advice. What advice do you want to give to the young research aspirants that they want to like establish their world into this antibiotic discovery?
It's a very personal thing people have to pick up actually, right?
Mm,
So what I would generally say is people should start focusing on thinking on precision medicine
mm,
first. uh, I give usually four mantras to it is we are all unique beings, right?
yes.
None of us are similar, so we have to
Yes.
respect ourselves first.
Mm,
And
mm,
second thing is we don't have everything that takes to achieve anything in this world, so we need to
mm,
collaborate.
yes.
And the third thing is acquire new knowledge as much as possible.
Yes.
And fourth one, always remember we are just a breath away.
Mm,
So be grounded and humble. Don't become arrogant with
yes.
the knowledge one acquires.
Yes.
So, what my advice would be in addition to this, technically, start with biology, not just chemistry.
Mm,
In our case, chemistry drove is good,
mm,
but
mm,
that's one. Second thing is embrace interdisciplinariness. Never say,
yeah.
see, though if you look at my profile, my friends actually from KMC, they ask me, every few years you change your hat. Why? I said, life is
Mm,
exciting. Others get
mm,
bored of. So I
yes.
like to learn new things. Imagine I was a campus director of Acharya Group, right? Where I
Ah,
was
ah,
heading 15 institutions, including engineering college, all the institutions. How that
mm,
was an exciting journey. I was
yes.
heading DNB program for 7
Mm,
clinical disciplines,
mm,
reading those theses, editing them, correcting them, new knowledge. So
mm,
interdisciplinariness broadens your horizon. So as a
yeah.
scientist, when you look at something, you don't have to do it for your lifetime. 2 2 years you do certain things,
Okay.
and then you
Mm,
acquire related fields so that you acquire
mm,
a broader way to not only do
mm,
science but also administration.
mm,
Right? That's why I said that. Then,
yes.
as I mentioned, we have to think precision medicine in the future, not just broad spectrum.
Mm,
Then already I mentioned about using AI ML tools because it's very
mm,
important. Because synthetic
mm,
biology, systems biology, omics, all these things come into that. We need to do that. Then keep resistance as a factor when you're designing.
mm,
How will bacteria fight back against this
mm,
molecule? Our question should
yeah.
be, how will my molecule fight the bacteria? No.
Mm,
how will the bacteria fight back
Yes.
this molecule?
Mm,
That is the question with which, when we start, we can prevent
mm,
resistance, right?
yes.
There are enough techniques to do that. Then
Mm,
we also have to understand the business and policy side here. We cannot just, oh, I get a molecule, but it is going to cost me, one dose will cost me 10,000, 20,000 rupees. No use.
mm,
So the
mm,
business aspect also has to be
yes.
put into play. Then
Mm,
team, team is important. We all have teams,
mm,
right? So
yes.
we have to ensure that the teams are science oriented. Everybody
Mm,
who walks into the office, you
mm,
have to forget your all your identity.
mm,
We are here as just pure scientists. Everyone is equal. No
mm,
one is superior to anybody.
yes.
No one is inferior to anybody. All of us
Mm,
are unique. There is no
yes.
equality. We are all unique.
Mm,
If equal
yes.
means there is everybody doing like a machine, same work. No,
Mm,
we are all
mm,
unique. We all bring a unique side. Collectively, we can make inventions or innovations happen. So that is very important to respect everyone's input or actions. So the
yes.
persistence is very important. And finally, we have to be mission driven. Distractions do
Mm,
come in.
yes.
Focus,
Yes.
perseverance,
Yes.
focus. You will
Yes.
hit where you want to hit, but
Yeah.
never get married to one molecule.
Yes.
It has to be shut down. We should have the
Okay.
courage. That's when business comes into
Yes.
play.
Mm,
Because many times I love this molecule. It's done well so far. I want to take
mm,
it to clinic, and you're just wasting your resources, but not making
yes.
headway. That's not done. So you
Mm,
have to stop at some point and say, okay, that's called fail early, fail fast. So
mm,
that's how I have made decisions in
mm,
eliminating so many molecules and take one molecule forward. And there's another program which we have done with US Merck where we could choose certain molecules to take it forward by this approach. We should know when to call it a day and
mm,
never I say I failed. I always say I
mm,
learned. When I say I failed, your brain thinks, oh,
yes.
but you say I learned,
That's
you
right.
say, ah,
Yes.
same thing. I learned one new way that, okay, if you do add this reagent in this, the reaction is like this. So I will not do
Mm,
this experiment. I learned that we should eliminate this from experiment. That's how you look at it. And that keeps you excited. Otherwise,
yes.
I cannot be this excited even after so many years, 18 years. Many people ask me, how did you sustain for 18 years with this molecule? I said, learn. Everything is a learning, never a failure. Every event is a learning. That
Yes.
makes your life happy and, go
Yes.
after it.
Yes. That keeps us going. Yes.
Absolutely.
Yes. It's thoroughly enjoyed the podcast session, sir. It was so nice to talk to you, and then how you have explained about the AMR, the emerging resistance, the newer antibiotics, and the clinical applications about it. And thank you so much for joining us for today's podcast session, sir.
You're welcome, Dr. Nischala. Pleasure that I could have this privilege of sharing whatever I knew with you, and always verify the information as well. Because I said it, it's not the ultimate thing. If something you have doubt, always cross-check the references and say, okay,
Yes.
maybe he made a mistake. That's okay, right?
Yeah.
That's good. Thank you so much for this lovely time we had together
Thank you so much, sir.
discussing
Thank
AMR.
you.
Bye
Thank
bye.
you. Bye bye, sir. Thank you.